All right, guys, so the rest of chapter 15 is in this video. We talked about how we transcribed and created our message in prokaryotes and eukaryotes. And now it is time to talk about what happens with that message. What do we do with it? So we said in prokaryotes, it's in the cytoplasm. In eukaryotes, it starts in the nucleus and needs to be transported out of the nucleus and into the cytoplasm to meet up with the ribosomes. So before it does that, it needs to be processed. So I like to think of it as a rough draft, making a paper. Here's your rough draft, and here is your final polished version that's so pretty that you're going to turn into your professor. So on your rough draft, you have two basics that need to be dealt with. You have exons and you have introns. You only want to keep the exons. And it sounds, normally I would be like exons, exit the system, but I can't. You keep the exons. Those are going to be the things that actually code. So the exons stay, and your introns get cut out with the spliceosome. So those are all going to get excised out with the spliceosome and dealt with. So exons stay, they are getting expressed, so that's why they have X on them, and the introns are going to be our in-between, intervening between the ones that actually get expressed. So introns go by and exons stay. Exons are going to come down, and now we have this pretty thing where we have all these different exons that we want to keep. But we said that prokaryotes, transcription and translation is happening within the cytoplasm. Prokaryotes, RNA only has to survive a couple seconds. I think it said in the textbook like 5 seconds tops. Eukaryotes has to survive longer. I think it said eukaryotes can survive up to a couple hours possibly. So to do that, we need to make sure that the RNA is not going to degrade in that time frame. And so how do we do that? There's two things that we need to add on to our RNA. And the first is the cap. If you like to think of formal attire, like a really formal black tie event, guys are going to wear their top hat and their tails with their tucks. So it's kind of the same thing. You have a cap and you have a tail. So both of these things are going to help prevent the RNA from degrading, being eaten up, and breaking apart in the cytoplasm by different enzymes and things that are out there. It's going to allow it to be stable. So we have the methylguanosine cap. Just call it the cap. It needs its cap, and that's going to go on the top, and the bottom is going to have this poly-A tail. Both of these allow for stability. The poly-A tail is also going to allow it to be, once it's got this tail on, it's going to help it get out of the nucleus and go into the cytoplasm. So these are things that happen in eukaryotes that prokaryotes don't have to deal with. We have to get rid of introns, splice them out, and we're going to keep the exons, and we have to add its cap and its tail. And now it's the final version that's pretty and ready to go out into the cytoplasm and meet up with a ribosome now. Trypanosoma, that is going to be a protist pathogen that gives you African sleeping sickness. And it is carried in a fly called a tsetse fly, and that fly bites you, and then it puts the protozoa into your body, and then you come down with African sleeping sickness. It's in this chapter because it, when it's creating its messenger RNA, it's missing some nucleotides. Instead of the T's, RNA has U's in its place, but it's missing some of its U's. So it does something that most of the other eukaryotes don't do when they process. It also has to do more RNA editing where it tacks in those U's instead. So they are going to have a poly-U tail and do some more editing. This is T. brucei, African sleeping sickness that you get from the bite of a tsetse fly. It has to do some more editing on its messenger RNA before it is a final polished version that will work with the ribosomes. Here is that spliceosome I was telling you about that cuts out the introns and gets rid of them. So it's just going to cut them out, and now we have nothing but exons in the end. And another video going through this to tell you how that happens if you want all the details. Moving on to the next section, when we talk about translating it, technically this is transfer RNA. This is in the previous section, but I'm going to pull it on down to the next section and we'll go over it there. So with this transfer RNA, it's going to have its anticodon on one end, and that's where it's going to meet up with the mRNA. And then it's going to have its amino acid site at the top. And so every single one of these is going to have an amino acid at the end, and each codon is going to link in with its own transfer RNA specific for that codon that specifies this amino acid that I'm carrying, and only I'm going to carry it because I have the specific codon. That is being carried by a molecule of RNA called the transfer RNA, or the tRNA. So there are three different types of RNA. There's messenger mRNA that gets transcribed. There's transfer RNA. That's another RNA that's going to meet up under translation. And then there's also ribosomal RNA, and that is going to create the pieces of ribosome that you see here. Here's our small ribosomal unit. We cannot have translation occur without both pieces of the ribosome. I am always thinking about food 24/7. To me, the ribosome looks like a hamburger bun. So we've got our bottom small unit of the ribosome, and it attaches onto the messenger RNA, but we can't proceed with translation without our top half, without our top part of the hamburger bun, our larger unit, the large subunit of the ribosome, and those are going to be where the transfer RNA binds with, and then it all comes together in this sandwich kind of event called translation. So here's our tRNA, here's going to be our amino acid at the end. It's coding, here's its anticodon down here, where it will attach on to the DNA and link in, the RNA, and link in codon by codon by codon by codon. When it carries over an amino acid, like this guy, we're going to have another transfer RNA that comes down right next to it in this large ribosomal subunit. And this amino acid dangling off the end is going to meet up with this amino acid, and they're going to bond. And remember, amino acid bonds are called peptide bonds between the two amino acids. Peptides for proteins, they both start with a P. Here's that peptide bond that's going to bind these two together, and eventually you're going to end up with that strand at the end of the primary protein structure. And then it went up to secondary and tertiary right when it got all complicated. So here's our peptide bond. Here's translation. We have the small subunit binding on to the messenger RNA. We have a unit of transfer RNA that is going to meet up with its large ribosomal subunits, and together they will sandwich together. We are going to have, remember, our start codon was this amino acid right here, and then we're going to bring in another transfer RNA and another one. So we're going to move codon by codon by codon by codon down the message and translate that message into individual amino acids that bind together with peptide bonds, and you end up at the end with a protein, a polypeptide. So this is translation, taking that message using transfer RNA in the ribosomes, and we will translate that message into a finished version of the protein. When it is finished, it's the same steps before: initiation, elongation, and then termination. What do I have this here? These are going to be initiation. We are going to have elongation. And then eventually we are going to have termination, where our protein is going to dissociate and we end up with our final finished protein. Some of these guys, remember we talked about primary, secondary, tertiary, quaternary structure. So they may be folding as they go, and they're not complete until they start to get their finished 3D version of that protein. Because remember, protein structure is important and it matters what the protein looks like. But these are the basic steps: initiation, elongation, termination. Understand transcription, translation. I kind of boiled it down a little bit. Then the textbook goes into a little more detail. But if you have questions about the basics about transcription and translation, please let me know. Here it's talking about the folding, that it may have to happen on these things. Usually it's going to have to happen. They don't look like a string. They're going to have some sort of folding going on in there to where it is the final version of the protein. But if you have questions, again, please email me or stop by into Zoom for office hours.